Hi, I'm Austin Circle, and this is an overview of our ESTCP project we worked on. It was project number ER19-5078. I was really fortunate to work on it, and I'm just going to jump right in with sort of the overarching goals and problems of this project. So the overarching goal of this project, as well as the related CERDIP effort, was to create and validate a method for the simultaneous analysis of 24 IM and legacy energetic compounds, in addition to surrogates, in environmental matrices, including waters, soils, and tissues. The goal of this project was validation of the method, which consisted of a series of round-robin studies, including participants from diverse scientific backgrounds, so including DoD federal labs, non-DoD federal labs, commercial labs, and academic labs. Both sample extraction and analysis were performed at the participating laboratories themselves, while sample spiking and homogenization occurred at the U.S. Army Engineer Research and Development Center prior to subsample distribution. The anticipated benefits of this method include enabling earlier identification of environmental concerns associated with the IM life cycle, and we expect this method to prove useful in areas where both legacy and IM are encountered, such as test and firing ranges, or production facilities. Moving on to results, all of our participating laboratories were able to successfully implement the method, and our statistical results, which I'll partially address on our next slide, showed comparable yet slightly wider recovery ranges in comparison to DoD QSM control limits for US EPA 8330B. For both aqueous and soil samples, lower analyte concentrations near regulatory limits were analyzed during follow-up studies, and the method's performance was satisfactory, even at these challenging analyte levels. We found that overall this method is highly capable. However, it is more difficult operationally and requires attentive analysts, preferably with some related experience with 8330B, HPLC, or SPE. This really emphasizes our end recommendation that this method isn't expected to replace 8330B, but rather to supplement it and be an option when simultaneous determination of legacy and IM is important. So to give a glimpse into what the end products of this project were, here's a comparison table of our direct injection data. So that's high analyte concentration in an aqueous sample matrix, with new analytes that aren't included in 8330B highlighted in these blue rows. You can see that our recovery ranges in the middle column are comparable to those of 8330B in the right column. These recovery ranges are also comparable to those found for EPA methods for other similar organic analytes by chromatographic methods, such as 8081 for pesticides, 8141 for organophosphorus, and 8270 for semi-volatile organics. All of our other extractions, so that's soils, tissues, and SPE extraction, were validated and performed well, with SPE extraction seeing slightly larger recovery ranges than we see here, likely owing to the increased difficulty of the extraction procedure. A follow-on study for soils was also conducted to compare analyte recoveries when soils were prepared with mortar and pestle grinding versus puck mill grinding. We found that performance between these two procedures is very similar. And lastly, all of these method validation results have been transferred to the US EPA for review and potential incorporation into 8330B as an addendum or option. This is the team that made this project possible. We'd also like to sincerely thank our round-robin participants for their help. After all, this really was a team effort. We also thank CERDIP and ESTCP and our project manager, Dr. Andrea Leeson, for their unwavering support of this project. Thanks.